Go ahead.
Hey, thank you, Gary. Yeah, this research is I'm going to be talking about today is part of my broader PhD research at the University of Amherst with Dr. Adrian Jordan, but very integral to the work I've been doing at DMF, and really the DMF's been doing for a long time with striped bass. So I'm pretty excited to talk to everybody today. And thanks for showing up. I just will say it's kind of a diverse group of co-authors on this paper, and it's been a real joy working with all of them on this. I'm pretty excited, so I'll get right into it, hopefully. All right, so as almost everybody here knows, striped bass are the premier saltwater sport fish for recreational anglers on the East Coast, and they do support important commercial fisheries in some states. So this is a tremendous resource for anglers, but can also easily lead to high degrees of mortality on this species. And as a diagenous fish, see, Gary, I don't get to leave diagamy behind yet. I have a complex life history that features a dependent, they have a complex life history that features a dependence on riverine and estuarine habitats. Now, historically, this has played out with a likely extirpation of populations from rivers that were dammed over the past centuries, but it's also meant that we can often predict the amount of fish we will see down the road from juvenile abundance surveys in important estuaries like the Chesapeake and Hudson. As is noted on this slide, we've seen a huge swing in striped bass abundance over the past 40 years as environmental conditions and regulatory frameworks have combined to push the abundance of striped bass both up and down. Now, as a diagenous migratory fish, we are presented with a management problem that is not uncommon, but has proven pretty thorny for striped bass. Although we know that fish caught in the ocean come from only a handful of populations at this point, we have not been able to account for the origins of striped bass. This means that as striped bass seasonally migrate from spawning estuaries in the mid-Atlantic to summer foraging grounds in New England and back again, they are being caught at a really high rate, but we have a minimal idea about the impacts to specific spawning populations. In another context, here's information from telemetered striped bass. We put out about 250 acoustic tags in 2015 and 2016. And you can see just how diverse the timing and the patterns are. But we have this broad seasonal migration in the summer in New England and in the fall back down the coast with movements up rivers into estuaries, overwintering off Delmarva. And then as we get into the spring, spawning migrations, entering those estuaries and all the spawning rivers. And it's an annual cycle, and there's variation of individual fish throughout that. So it's very complex. So despite not having the data we would like to have to manage these fish, striped bass have been considered somewhat of a modern success story. Although the shine on the apple has certainly faded since the late 2000s, mostly due to poor recruitment, especially in the Chesapeake Bay. As we saw in the earlier figure, and again here, of spawning stock biomass and recruitment, there's certainly a relationship between the two, but it seems like that all we know about striped bass's life history and the historical record point to this being a mostly one-way relationship where recruitment predicts spawning stock biomass, but not vice versa. This is complicated given that most of our management metrics revolve around spawning stock biomass, which is certainly a decent goal, but it does not seem to guarantee the returns that we want to see. Given this reality, plus the ever-growing human coastal populations impacting estuarine nurseries and added stressors from global climate change, management models that incorporate resiliency, like this one adapted from a 2016 paper by Susan Laura Barbieri et al., make increasing sense. So, as well as contributing to data deficiencies in the striped bass stock assessment, I would say that we should also be thinking about making the tools and collecting the data that will help us manage for resiliency in this population. With these ideas in mind, myself and my collaborators have been enacting a research program to bring rapidly developing fisheries tools and techniques to bear on our understanding of striped bass in the hopes that it will lead to a better management strategies at coastal and potentially more local levels. The first goal relies primarily on acoustic telemetry, the second on genetics, and the third on a combination of both. But today I will be largely focusing on the second of these goals, although I will briefly touch on the first as well. So, as a reflection of the importance of this mixed stock problem, many folks have tried many different ways to assign coastally caught striped bass to specific populations. This 2012 review paper by Waldman et al. Does a really wonderful job of summing things up, but since then there have been several new efforts, especially in the realm of genetic markers. And this work has tracked the advancement of genetics through microsatellites into SNPs, or single nucleotide polymorphisms, in an era of genomics. While SNPs and microsatellites have been developed or used for genetic stock identification of striped bass over the past 5 years, the current panels do have some important shortfalls. In the case of microsatellites, the process of getting reproducible results among different labs is really difficult and not guaranteed, making any large-scale implementation of a monitoring program doubtful. Further, microsatellite panels are typically quite small and can have substantially less power than SNP panels. On the other hand, SNP panels can deliver very powerful results, but at a substantial cost if a panel relies on thousands of markers, as can often be the case. So I don't often read directly from my slides, but when I do, it is because I think it's important. The goal of our project was to develop an accurate, precise, standardizable between labs, and cost-effective method to determine mixture estimates of potentially individual assignments of striped bass. And to do this, we started with the DD-RAD panel we published in 2020, and we looked for a smaller subset of loci that would be the most informative for GSI purposes. So as a reminder, as with other SNP and microsatellite efforts, we determined from our set of reference specimens that we could distinguish a 6 total population or groupings of populations, and I'll refer to these as reporting units from now on. And so hopefully my mouse will show up, but these were kind of a Gulf of St. Lawrence population or reporting unit, the Shubenacadie in the Minas Basin, the St. John River in the Bay of Fundy, then a Kennebec Hudson reporting unit, and a Delaware Chesapeake reporting unit, and finally, Carolina reporting unit. So this is an output from a program called Structure. And for folks who haven't seen it before, you can kind of see how these are grouped by colors. Basically, each tiny vertical strand of this figure represents one fish. And then the colors are the assignment of that fish over thousands of runs of simulations. And so when you see a very cohesive color, it means that most of these fish were all getting assigned back to that population almost every run. When you get into the US stocks and you start seeing this mixed band, it means that there was a lot more variability in where a single fish was assigned over the course of thousands of simulations. And that's a little easier to visualize in this discrimination analysis of principal components plot, showing here these are the 3 Canadian reporting units, and then in 2-dimensional space, and then there's the United States. So we knew they were separating in structure. We were seeing population structure signs in our 1200 SNP panel, but we did not know how well it would perform once we started winnowing down those SNP markers. But running, each fish at 1200 SNPs meant that we were going to be looking at roughly $50 per sample, which was a little bit economically prohibitive to do much with. So, despite these kind of concerns and the lack of differentiating that we saw in that figure, we were very, very happy to see that the 233 SNP panel did very, very well. I'm going to show a few things about how this is performing. This is kind of a whole separate paper, but this figure is based on some simulation work we've done. And we were interested to see what sample sizes we needed to have if we want to do mixed stock estimates up and down the coast. And so here you can see a scenario over 2,000 runs of simulations with a 80% proportion of Delaware Chesapeake fish on top, a 20% Kennebec Hudson. The colored symbol is the mean of the distribution. The black X is the median, and then the bars are the 95% intervals on that. And so those are for the Delaware Chesapeake and the Kennebec Hudson. And you can these gray, the gray points next to them are just for the random sampling error you might expect over the same number of simulations, and sample sizes on the bottom. And what you can see is that somewhere between 200 to 300 samples, both of these stabilize really well, and you're pretty much just at a point where you're returning the same thing you would get from random sampling error. And that was very, very promising and in line with some of the work that's been done on other species with SNP marker panels. So that was for mixture estimates. When we looked at individual assignments, it also performed very well. This is a figure showing the posterior probability of assignment for about 250 fish. Some of them are left off. You can see probability of assignment on the y-axis and the tagging aggregation. This is part of a acoustic telemetry study. I'll get into a little bit more depth following this, where we tagged in 3 different locations. And these are box plots, but they just pretty much just see the outliers coming down. But the takeaway here is that almost all samples had a greater than 90% probability of assignment to an individual population, or reporting unit. So this performed really, really well. We're really excited about that for reasons that'll come clear later. And then hopefully, most people are familiar with this matrix showing where the accuracy of what is considered the accuracy of assignments of the reference panel. So these baseline fish that were collected in all the different spawning areas. And you can see for the most part, almost all the fish are being assigned back to where they were collected. That's these numbers in bold black. And then the red numbers are what we would consider potentially misassigned fish. And then on the end, there's that column for unknown. And those are all the fish that had less than 90% probability of assignment. So we just classified those as unknown. And by extension, these fish that are quote unquote misassigned, these are all fish that have strong genetic support that they are not in fact being misassigned. They are actually from the reference population that they've been assigned to, which raises the question about do we now have the ability to better understand phylopatry in striped bass, which has been a topic that a lot of people have talked a lot about, but we've not had a lot of data to support. So I'm going to make a kind of a short digression here into the telemetry work we did. To address that first goal I presented earlier in the talk, we did a 250 fish telemetry study that we tagged fish in 2015 and 2016. And you can see these 3 areas on the right where we tagged the fish. And based on all the telemetry results from those fish, we were able to assign spawning areas. So I considered a fish to be spawning in an area if they were in a known spawning location in the correct time period. For the Hudson River, that meant one of the 2 main areas identified through seine surveys and egg sampling. And there's a really nice figure in Secor et al. 2020 showing that. So that's Bear Mountain Bridge and up. We're considered to be spawning fish. For the Delaware River, it was above 39.5 degrees latitude. And Chesapeake, just because there's less coverage, I was a little bit more liberal in saying if it was in the Chesapeake Bay or any tributaries during the spawning period, I said that it was potentially spawning there. So this is a chord diagram. And for those of you who are not familiar with the chord diagram, the way it works is on top here, we have the tagging aggregation. So where we tagged fish, Boston Harbor, Cape Cod, Buzzards Bay, Vineyard Sound, and then where they were eventually detected spawning. And we have the Hudson, Delaware, and then this is fish that in some years or within the same year visited both the Delaware and Chesapeake spawning areas in the right time period. And then these are fish that were just detected in the Chesapeake. These 2 fish I've identified as quote unquote strays or vagrants are fish that in 2 or more separate years were in the Hudson and either the Delaware or Chesapeake spawning areas at the right time. So seem to be making a larger switch than just the Chesapeake Delaware reporting unit in multiple years. And you can see that Buzzards Bay seems to have a different composition, but Cape Cod and Boston Harbor are largely the same. As a note, we tagged about 250 fish. We only had about 105 fish that we had spawning locations for. This is likely due to us tagging a lot of smaller fish that may have not been spawning yet and are spending the spawning period either lower in the estuaries or in the coastal areas foraging. So the nice part is that with the genetics, we can now look at the same idea, aggregation on top again, but now reporting unit. And it's different. What we see is a lot more Delaware Chesapeake contribution to these aggregations than we saw with the Kennebec Hudson. You can see also there's a single North Carolina fish that we had tagged. as well as two fish that I really appear to be admixtures between the Hudson and the Delaware Chesapeake. So, and again, this is at more than 90% probability of assignment. So maybe it's the small fish I mentioned that we didn't get detections of in a spawning area, but could it also be behavioral? So we can turn it around and now have ACTEL spawning, our acoustic telemetry spawning on top, and our reporting unit genetic assignment below. And what we see is that there is heavy, heavy Delaware Chesapeake contribution even to all these spawning areas. It makes sense for Chesapeake, Chesapeake, and Delaware. But roughly 20% of the fish detected on Hudson spawning grounds in the spawning season had the genetic signature of a Delaware Chesapeake origin fish. We can't know for sure that they were actually spawning. Obviously, there's large migrations of shad and river herring going up and other forage fish going up the Hudson at the same time. These could be fish that are just in the river feeding, but they were on the spawning grounds at the correct time. So I'm going to say these were spawning for the sake of argument. So my basic point here is that we have seen evidence of strain in our acoustic telemetry results, in our baseline, and in the fish where the genetic origin of our acoustic telemetry fish. So we're seeing here is actually behavior, perhaps this strain behavior being more common than sometimes thought, and a potentially important resilience strategy for striped bass. So digression away, I'm going to move on to the meat of the topic for today. And I'm going to start by presenting some of the details about our available samples, genetic samples for this study. So to prove the utility of the panel, we applied it to a nearly 5,000 sample data set collected primarily in Massachusetts. All the Massachusetts samples came from our collection programs that DMF already conducts on an annual basis. So it really illustrates how future genetic monitoring programs could easily be incorporated into the current agency activities. A lot of other states are doing a lot of the same sampling. So hopefully it would be an easy lift if we can get a pathway forward for that. So out of all our available samples, we then randomly selected these 5,000 from the available pool of samples based on questions we had about annual composition, the area collected, and the regulations in place in Massachusetts at the time the study began. So that led to more than 250 samples in every area and year combination. And, sample sizes did differ depending on the question we asked. For instance, sample sizes were adequate for two-way interactions, but not three-way interactions. So I'm going to continue to provide these graphics just so everybody can see sample sizes throughout the results. I do also want to note that we ran a smaller set of samples from Connecticut waters to help get an idea of how areas outside of Massachusetts might perform. So we use the program Rubius to perform all of our GSI analysis. Rubius uses Markov chain Monte Carlo methods to determine individual assignments and mixture estimates. And we found that the default settings delivered stable and reliable results while also minimizing computation time, with the exception that we did use that parametric bootstrap analysis for our mixture estimates because we saw a slight upward bias in mixture estimates towards the Delaware Chesapeake. And to determine the differences in composition in the estimates, we compared the distribution of the estimates over the final 1,800 of 2,000 sweeps of the MCMC chain. So the matrix on the right here is a way of visualizing how the differences were determined. So the red hued squares show where distributions overlap more than 5% of the time. So for instance, this is the Delaware Chesapeake composition of Buzzards Bay Vineyard Sound, and this dotted line is the mean. And so with Cape Cod, you can see that the distribution does not overlap the mean at all. But with the Connecticut samples, you can see that more than 5% of the tail of this distribution overlapped the mean Buzzard Bay Vineyard Sound. So this is really a way of saying like a 95% probability, as most people, not statisticians, understand it. 95% of the time, if we say something's different, it was different. So hopefully that makes sense. It's a little complicated. I do want to note that Carolina fish were present in almost all of this mixtures, but they were always at very, very low levels. So a lot of the times I'm not going to speak directly to Carolina fish. So with that, I'll move into results. Here's collection area composition. This will be the only results I show with Connecticut involved, but I think it is useful to take a quick look at those. So here's our sample sizes. Again, Connecticut has an order of magnitude less samples than the rest, and it occurred in a different time period. So take that into account. So the estimates for rep unit composition by region from north to south are along the the x-axis, and on the y-axis is the proportion of the mixed sample with Carolina as purple, Delaware Chesapeake as teal, and Ken Hudson as yellow. The black bars are 95% credible intervals from the 1900 post burn-in sweeps. And as was illustrated by the matrix in the previous slide, these are all three mass regions were composed of more than 70% Chesapeake Delaware bass. I mean, there were differences among them with Buzzards Bay Vineyard Sound having the highest proportion of Delaware Chesapeake and Cape Cod the lowest. And Connecticut was composed of a similar blend as northern Massachusetts and Buzzards Bay Vineyard Sound, which could be due to geographic sorting, ontogenetic progression, or the timing of the collection. A lot of questions remain there with the smaller sample size. Moving to annual trends across size classes and years, our sampling was relatively even and revealed what appears to be a more stable pattern. We see in 2018 that the highest levels of Delaware Chesapeake reporting unit occurred, although they were not a great deal higher than the rest of the years. One thing that really jumped out in my mind when I saw this is whether that was a recruitment pulse from the 2015 Chesapeake Bay year class that's driven a lot of the recent management changes for striped bass. And thankfully, we can drill down on this question by looking at some of the two-way interactions. So first, we can take a look if the pattern we observed across all areas combined holds true once we look at individual aggregation areas. And again, on the left, you can find the sample sizes by area, and note that even with the two-way design, most samples are nearly at 300 fish or above. And you saw from that simulation mixture that produces very tight, credible intervals and good, precise estimates of composition. So the data here are in a slightly different format with both colors and symbols being important now. As before, we have Carolina in purple, Delaware Chesapeake in teal, and Ken Hudson in yellow. But now the symbols are Buzzards Bay Vineyard Sound as a cross, Cape Cod as a circle, and northern Massachusetts as a triangle. We have year on the x-axis and the proportion of the sample on the y-axis, and the black bars are the credible intervals. And again, I did not show the differences with letter characterizations, but you can basically see if you can visually tell if those credible intervals are overlapping a mean or not. And the takeaway here seems to be that it's pretty similar to the earlier pattern. Cape Cod and northern Massachusetts were not different from each other in most years. Well, Buzzards Bay Vineyard Sound was. The exception, of course, is 2015, where northern Massachusetts was more similar to Buzzards Bay Vineyard Sound than Cape Cod. And we do see a dip in Kennebec Hudson and rise in Delaware Chesapeake in 2018, which again could have to do with the 2015 year class, but it drops after that to lower levels in 2019, especially for northern Mass and Cape Cod. So this was kind of an inconclusive trail that I went down. So next I looked at size class by year, and you can see the different size classes and the samples for each year. And again, we have good sample size, except with maybe recreational in 2019 that was below 200, but most combinations had really strong sample sizes. And also you can see that the most numerous samples are those school size fish. So it's good to think about as we look at all our results that our pool of samples is slightly weighted towards smaller fish. So same type of plot as the previous slide. But here the symbology changes. Crosses are commercial size fish, circles are recreational size fish, and the triangles are school size fish. And instead of seeing a large surge of school size Delaware Chesapeake fish in 2018, we can see that the larger gains were actually in the commercial and recreational size classes that we sampled. So overall, the pattern of that school size composition does most closely resemble the pattern and of like the overall sample. and displays a steady rise through this whole time period of sampling, while commercial and recreational compositions showed greater interannual variation, while also being very similar to each other. The commercial and recreational fish were very similar to each other from 2016 through 2019. So kind of the takeaway regarding the 2015 year class on this slide was that it doesn't seem like the culprit for that rise in 2018, although it is possible that the overall rise from 2016 onward in Delaware Chesapeake Delaware Chesapeake reporting unit fish may be related to that cohort. That would be an interesting idea. It's a kind of a different take than we know from telemetry about the emigration of these fish. What we do seem to know would be that there's a steep break around 80 centimeters where almost all of the fish depart Chesapeake Bay, whether they're male or female. And that was a recent paper released by Dave Secor and co-authors. And in the Hudson River, we definitely observed smaller fish joining the coastal stock. But it seems kind of likely, given what we're seeing here, that telemetry techniques have yet to really adequately capture the full suite of contingent behaviors and their downstream impacts for populations. So genetics in combination with telemetry or hard part microchemistry should offer paths forward in the future. So that was a few slides about what we can do with the mixture estimates. I'm going to move on to individual assignments. And this is really exciting stuff, I hope, to me. So I hope everybody else thinks it's kind of cool. So it's really neat what we can do with individual assignments, because now we're talking about individual fish, and we can combine them with biological data or any other data type we have on those individuals. I'm going to do mostly explore age here. And just as a note, over half of the fish in these analyses were aged directly by DMF's Age and Growth Lab. So thank you, Scott Elzy, and everybody who is currently in or has worked in the Age and Growth Lab. It's a fantastic resource to have. So the remaining samples I had to assign ages from your specific age length keys. Thank you, Gary, for passing those on. And I used the semi-random method from Eiserman and Knight 2005 in the R FSA package to do that. So on the left here, you can see reporting unit proportions by age of all fish, with peak sample sizes at ages 4 through 6, and then decreasing to a handful of striped bass as we get all the way out to 20-year-old individuals. I mean, really, I am going to present the 18, 19, 20-year-old age classes, but take those with a huge grain of salt, given really the 17 through 20, how low the sample sizes are. But speaking again to the capabilities of the GT-Seq panel, to kind of bring people back around that, we only lost 134 samples to low probability of assignment. That's below 0.9. So this left us with many, many, many samples to ask some very interesting questions of the data. So here are the reporting unit proportions by age. Interestingly, the Chesapeake has some of the highest levels of contribution at the youngest ages. And the older ages make sense, as the numeric dominance of the Chesapeake population would win out over 2 decades of mortality. But given the much greater migration distance, a greater proportion of Hudson fish at the youngest ages is more of what I was expecting. So this brings up some great potential ontogenetic questions, but of course this plot is influenced by our sampling representing a 5-year period in time from 2015 through 2019. Which is why it is informative to look at the year classes that we have the data for. So across our 5-year sampling period, we collected fish from 21 different year classes between 1997 and 2017. And as might be expected, samples were most numerous for the year classes between 2005 and 2015, as older year classes had suffered a decade plus of mortality, and some of the younger year classes were likely still recruiting into the fishery. So looking at the composition of the year classes, we see a slightly different pattern than the mirror of age, but mostly there are similarities. As with age, the oldest surviving year classes are made up of mostly Delaware Chesapeake fish, and so are the most recent cohorts. This is especially interesting given that outside of 2011 and 2015, recruitment in the Chesapeake Bay has been mostly poor, yet the proportion of Chesapeake fish has increased over that same time period. So we can also look at the aggregation area to examine potential differences within state waters. And here we see largely consistent overall patterns, but slight differences, especially in the Buzzards Bay Vineyard Sound area, where, as we saw in some of the mixture estimates, Delaware Chesapeake fish appear more common than Kennebec Hudson ones do. So between the overall pattern and what we're seeing when we drill down to areas, Again, we're starting to maybe point towards ontogenetic differences in migration timing, in how far fish go based on their age or size. If they, progressively go further north over the course of their life, these are the things that we're starting to see as I am really, just looking at the data in the early phases. So if we can do that, can we track cohorts? Yes, we can. So population-specific cohort tracking, especially when expanded to catch, these are not expanded to catch. This is just proportions of our samples. These are fisheries-dependent samples. We tried very hard to, sample evenly across these size classes, which gave us good age classes as well. So these are, I think they are representative of what are being caught in the fishery. But we still need to expand them out to catch. But that's going to be the point here, is that we have the tools to do it and we have the data to do it. So we only have a relatively short time span over which we sampled, but the presence of cohorts moving through is really evident, hopefully, to anyone looking at this. For the Chesapeake, the 2011 and 2015 year classes are evident, as are some of those remaining large year classes from 2005 and prior. And in the Hudson, large year classes from 2007, 2010, and 2014-15 appear in the same sample pool. These estimates are the first available for populations or groups of populations for striped bass, and when paired with catch estimates, really should allow for major improvements to estimating the fishing mortality of striped bass and the performance of, hopefully, of the assessment. Moreover, if we can tie these estimates to the number of fish that have and have not yet joined the coastal stock, we can continue to really drill down on the potential endogenous and exogenous spurs for migration, especially if we can pair the data with further biological, behavioral, or genetic, like potential adaptive genes or gene complexes information at individual and population levels. We can also examine how changes to regulations like slot limits to protect larger fish end up playing out in our fisheries and on our populations. So finally, this is my final data slide, and this is really preliminary. I was doing this yesterday afternoon. Since cohorts were evident, I really wanted to bring in recruitment indices. And as before, the proportion of samples is represented by the bars, and the red line here is the bay-wide. For Delaware Chesapeake, it's the bay-wide geometric mean from the Maryland DNR survey, and in the Hudson is the New York DEC SANE survey. So these are all proportions, and the SANE survey, both proportions and the SANE surveys were standardized to a 0-to-1 scale. Since Hadley does not believe in secondary y-axes, I had to do that, but I think it's also very useful, of course, to look at the data on similar scales. The gray rectangles on the right are just a reminder of the collection period, and that we have few fish from the older year classes left and incomplete year recruitment from recent year classes. So there's also obviously noise in recruitment indices, and we likely have increased aging error from the assignments of ages via an age length key for about half the samples. So despite all that noise, all those caveats, I think that there is a trend here that you can see in where recruitment seems to be our fisheries sampling reflects the recruitment indices that we have in our major estuaries. So this is really promising for future analyses, and it really points to the utility, again, of developing ongoing data collections along the coast to supplement what we already do for the striped bass stock assessment. So at this point, I've had the data for about 3 months, and I feel like I'm still breaking the surface of what I can look at and what the rest of my collaborators and I can look at. The stock assessment implications are promising. I can't wait to be linking age and year class patterns with catch estimates. In the meantime, I hope that I was able to kind of communicate our enthusiasm with the performance of the panel, and also leave you with some results that help us better understand some of the stock dynamics playing out in our coastal waters. And while the overall dominance of the Delaware Chesapeake reporting unit is notable, managers may be able to eventually use variation in reporting unit composition, especially if we can couple it with ecological and behavioral information to react to changes in population levels while preserving opportunity as much as possible. And that could be definitely through more refined spatiotemporal management. It's also really important to remember that, while we're sitting here saying, oh, Delaware Chesapeake is driving the train on one hand, that smaller unit, the Kennebec Hudson unit, small changes in overall mortality on the coastal stock could lead to larger changes on these smaller populations like Carolina or the Kennebec Hudson reporting group. And we always have to keep that in mind. On the other hand, I think that for folks who are active in management and have, looking at these results and really seeing the dominance of the Chesapeake, the Delaware Chesapeake reporting unit to the Massachusetts fisheries and perhaps other coastal waters, the concern of that recruitment failure that's ongoing in Chesapeake should be taken incredibly seriously. Coast-wide sampling and sequencing of these patterns can provide fisheries managers with the tools to better monitor the abundances of spawning populations. We were able to get DNA from scales and fin clips. I didn't mention that earlier. We're very successful doing that, which means we can also go back into archive samples and run these analyses backward in time to better understand what has actually happened over the past 30 years. We have a bunch of different estimates from different methods. We could actually come to concordance on that. So it's going to require an investment, but the gains will be very measurable, I feel, and will give us a better chance of reacting to changes in populations levels of striped bass while preserving as much angling opportunity as we can as we move forward. So I just did a big information dump. I just want to say thank you, a very, very, very sincere thank you to everyone on this slide and everyone at Division of Marine Fisheries. I've received a tremendous amount of help and support during my time working on this striped bass research, and science is never accomplished alone. I feel really fortunate to have wonderful, passionate, and capable coworkers and collaborators. A huge shout out to the University of New Brunswick St. John laboratory technicians for doing all of the lab work around these 5,000 samples. It is a huge lift, super appreciative. And with that, I'm happy to take, and the funding for this, I do want to point out, sorry, came from the DMF recreational license sales. So thank you to all recreational license holders, Sport Fish Restoration Program, and the ACCSP. So I want to say thank you to the funding sources, and I'll take questions.
Thanks, Ben. Questions for Ben? You want to come over here?
Yeah, Dave.
I guess you're not going to see me. But Ben, that was a great presentation. And I think one of the findings I came away from this was that Merriman was right. You know, that when you have a big year class in the Chesapeake, you start seeing all these small fish all over the place. But it definitely goes counter to our study with the telemetry. And it suggests to me that we might have not tagged small enough fish, that there might be two groups of fish that leave the Chesapeake, fish that are really young, maybe age zeros, maybe age ones, and then another group that we saw, because we tagged 15, 18-inch fish, and we saw them grow into that migratory stage. So we might have a really young kind of migratory group that we're just missing. Could you comment on that?
Yeah, Dave, thank you for that great observation. I think you did a much better job of articulating what I was trying to earlier, where I've been lucky enough to collaborate on a lot of these studies and work on them with you. And yeah, I think that that's absolutely what's going on. You know, the limitations of the tagging technology in the past, the tags are getting smaller all the time, which is great, but then you're dealing with trade-offs between tag life and detection range. It really appears like Merriman was right. We are seeing large pulses of very small striped bass from both, all the major spawning populations entering coastal waters at small sizes. I did, at a request of you, look at fish under, I think it was under 15 inches that we had, and there's only about 300 samples, but those were, I can't remember the exact number I sent you last night, but roughly 80% Delaware Chesapeake. So these are small fish that are making their way up to Massachusetts as 1 and 2 year olds. It's, I think, a really underappreciated part of contingent behavior. And the cool thing is that we're starting to get the tools to ask more. So again, I think that this is a situation where whether it's genetics alone, genetics with smaller tags, or with hard part microchemistry to kind of understand the ontogeny of these early migrants and what, and then also, as I pointed out in a different slide, kind of if we can figure out the relative amounts of these fish that are leaving early as opposed to staying and migrating later or staying their entire life in their natal estuaries, it's going to be incredibly useful to figure out how those topics that we've talked about a lot, their portfolio effects and life history diversity, influence the abundance of striped bass and the resiliency of striped bass.
Mike Armstrong has a question.
Hey Ben, this is just fabulous stuff. As someone who sits on the striped bass board, I just see so much potential that's going to help with management. Um, One question. So I looked at 27, sorry, 2012 out of the bay was a real clunker, maybe close to the lowest in the 70-year time series, and yet it comes out pretty strong. That's one of the anomalous points. One explanation, I'll throw out mine and then you'll give me the real answer, is that we have a capability of hitting weak year classes strongly, even if they're weak, which is pretty disturbing. And that's a battle we fight all the time. But do you have any other ideas?
I do not, Mike. I think that's a really interesting perspective, and I in the concurrent time, the unfortunately the Hudson was also not doing great in that same time period. And here's that 2012 point you're talking about, but yet it shows up. I can say that, in the time period that we were sampling, and you can see that in some of the sampling sizes, if I go back to sorry, to here, is that we got to 2019, and although we did have school size bass, we had trouble. We have less samples for 2019 when some of these fish were really like that 2012 fish would be, your class would be, 7 years old at that point, and is getting into that recreational slot limit. We had a hard time getting samples, and that's with our full sad cat sampling program where we're getting thousands of samples a year sometimes. So, it's difficult. So I think it's likely that where, this is proportion, it's not amount. So that's part of what's going on there. But, anglers are going to go out, they're going to fish. I talked a lot about recruitment being a big driver for why we're concerned about striped bass right now. The kind of outside of the topics I was covering today is, of course, fishing effort, which is the second thing that is driving a lot of our management efforts right now. And I think it shows up exactly in that point you just made.
Thanks, Ben.
Other questions, either unmute yourself or put them in the chat.
I have a few questions, and they're more like what you meant by Delaware, Chesapeake, and Kennebec Hudson. So when you say that, do you mean fish that spawn in the range from the Chesapeake to the Delaware, or like Chesapeake and Delaware? And the same thing for the Hudson to the Kennebec.
Yeah. So what I mean is that we have reference samples, so spawning fish from those rivers, and we believe those to be, we believe that there's not spawning to any significant or real amount in other rivers between those. Genetically, we can't tell them apart. In the case of the Delaware and Chesapeake, that's been a ongoing result with across different marker types and different panels of markers. And what we've seen in a lot of telemetry papers and studies is that we have fish that go between the two during spawning season. The thought there is that the C&D canal, the Chesapeake Delaware Canal, which runs basically from the head of bay spawning area in the Chesapeake straight into the spawning area in the Delaware River, the lower part of it, has enabled a lot of strain between those two populations and integration between the two populations to the point where they are pretty much look the same genetically now. And that could be in large part due to the real near collapse of the Delaware and then kind of getting relit by Chesapeake fish, or just there being that much changeover between our integration and swap strain between the two. In the case of the Kennebec and Hudson, the Kennebec is a recent restocking of fish. It was an extirpated population. Dam removals have allowed that population to be restocked, and those fish came from the Hudson. So they genetically look similar. As we move forward, we saw with the Atlantic salmon program that eventually those fish did start to separate genetically. So we might see that with the Kennebec and Hudson populations, but right now they look the same genetically. Does that answer your question, Dylan?
Yeah, it absolutely does. Thank you. Do you think that the anomalous year class thing that could be as a result of Delaware fish contributing at a higher level than Chesapeake fish in that year, that 2012 class?
It's possible. I don't think that the Delaware, Gary, you could step in here a little bit since you're far more versed in the power of the recruitment indices, but I don't think Delaware is ever really much of a contributor to that combined index of recruitment. Sorry, it's certainly possible, but I think it's more likely that it is that whole where my slides are all based on proportions, so it kind of looks like a lot more than it actually is if you were to factor in catch and then actually get to true amounts.
Right, I got you. Thank you.
Yep.
Just to let you know, in the stock assessment model, the Chesapeake, Maryland, Virginia index is usually the strongest index in the whole model. Delaware does show some similar recruitment trends, but the Maryland, Virginia seems to be highly correlated with the catches and everything. Oop, next, I think Tara has a question.
Hey Ben, amazing talk, really interesting results.
Thanks, Sarah.
Sure. Given what you just said about the mixture between the Delaware and Chesapeake, I think you had a few slides where you showed that there was some potential straying. It seemed like a high amount. Was it like 20% between the Delaware, Chesapeake, and the Hudson Bay, Kennebec? Given those high rates of straying, it's really impressive that you're able to do these to differentiate these mixtures. So could it be that the straying individuals are straying only recently, or they're not spawning in these populations? Can you explain that a little more?
That's something, Tara, that's a great observation, and it's something I've been puzzling myself about, and I'm not as strong enough. I like my throwaway line is kind of like, I'm not a geneticist, I just play one during talks. But I am, something that is certainly interesting. I've gotten the same question, and it's, I don't have a solid answer for that right now. It's intriguing that there might be that much strain happening, yet we still are seeing enough separation genetically to do such a good job assigning fish back and coming up with mixture estimates. Or like you said, it could be a temporal thing. And I think that some of these things, somebody who's a little bit more knowledgeable about the factors that influence our ability to discriminate among stocks could help answer. But I do also think that part of it is that temporal aspect you're bringing up. And again, so outside of what I'm trying to do right now, but in the future, I'd love to see a large scale effort of archive materials happen. And maybe we could start to look at questions like that. We could find a way to look at questions like that. It'd be hard to do without some of the telemetry results, but if we have any type of archive samples from spawning grounds, we should be able to do it. So these are the kind of questions I think that, the other part about this is just being able to really go back, and we have such great data in many ways for striped bass for such a long period of time. And we can augment it very powerfully to understand what has happened and hopefully help us do a better job in the future.
Cool. Thanks, Ben.
There's a question in the chat. Dave Secor asked, where'd it go? Was straying sex biased?
Dave, of course, you have to find my disappointments. Being that we tagged fish on not on the spawning grounds, but up in Massachusetts, and we were unfortunately unable to find a genetic marker for sex determination, or sex, not determination, not in like the sense of what is determining the sex of an individual, but determining what sex the individual is. We don't have an answer for that. I wish I did, but there's been few disappointments so far in this project, but that is one of them. So I apologize.
Elise Koob has a question.
Hi, Ben. I was just curious. I think it's the plot right before this when you have the tagged versus what you assigned the spawning areas to. Was that just looking at the next year of tagging data or multiple years?
Next year, if there was only 1 year of history, and multiple years if there were multiple years of history. You know, similar to what's been seen in many other studies, we had an attrition rate of 30 to 40% in any given year. So, I mean, you put 250 tags out and you feel great about it. In our case, we put out, you know, 150, then 100 roughly over 2 years. But in reality, you lose fish so quickly, plus fish that you didn't have, that didn't go anywhere, maybe because they weren't just part of a spot, you know, weren't sexually mature yet. So we only have determinations for 105, you know, less than half of our fish, which is kind of a bummer, but it's what
The
it is.
ones that you have multiple years for, did they always go back to the same spawning?
No, sorry. So like that's like this stray one,
Oh, okay.
and a lot of these Del Chess fit ones, like those were typically. I think one of these Del Chess fish was the same year. Went in both. Like was up in the, I think it was in the Potomac, and then jumped out and then into the Delaware for a couple of weeks and spent like 3 weeks in each area. But both of these stray fish were, you know, 1 year in the Hudson and 1 year in the Delaware or Chesapeake. And then there were other fish that had multiple years in the same location.
Okay, cool. Thanks.
Yep.
Other questions?
Gary, Khalil here. I have a question, please.
Go ahead.
Ben, awesome presentation. A lot of complex information in there. This is more of a practical thing. Back around 2014, 15, we were catching, recreational anglers were catching a lot of fish in the 6 to 9 inch range, which was highly unusual. Could you have any thoughts or any information about that particular group of fish, that why we saw those in Essex River, Plum Island Sound?
Yeah. And it's a good question. And so if you go back to, was it here? Oh, sorry. Wait, I've got to think about where it was for half a second. It was in the baseline. I don't know where it was. I apologize, but no, it was here. Okay, yes. So if you look down here, Khalil, you can see this HUD 12, HUD 14, Del 2012, Del 2014. The actual origin of all this work was those fish you're talking about, where all of a sudden 2012 and 2014 we had these pulses of really small fish on the North Shore of Massachusetts that, like, you know, very, very small fish, and we're trying to figure out where those things were coming from. And so we started collecting baselines, and we did some initial microsatellite type work, microsatellite marker work that evolved into this SNP project. And yeah, those you know, different marker set we have a lot less confidence in, but they were showing in one of the 2 years that it was, if I'm thinking back correctly now, that it was more Hudson, and one of the 2 years it was more Chesapeake. But yeah, really amazing and interesting thing when you start catching a 9-inch striped bass in you know, in the north, lots and lots of them on the North Shore of Massachusetts, because you're pretty far away from anywhere you'd think be spitting those out.
Yeah, it's interesting because a lot of people came up with a lot of different theories or hypotheses that, you know, their natal waters were other than what we traditionally think they are, and then you have the whole phenomenon of the Miramichi up in Canada, the striped bass population up there. Any thoughts on that one?
Yeah, totally. And I mean, I think that goes, I wish I had spent more time on this figure here, but that kind of goes to like the complex life history of striped bass. The genetics work we've done, like doing the diagnostics for the genetics panel, suggests that we didn't miss any populations. So to the first part of your question, it doesn't look like we're missing anything genetically. So there could be a spawning in a river that's contributing to one of these reporting groups that we don't know about, but the genetics don't seem to be like, it doesn't look like we have fish that don't belong to any of our reference populations. And then there's this idea of, like, I'm trying to think about where it would be in here, but foraging habitat and connectivity among populations, I guess, is not as much into this one as I thought it was going to be, but there can be, barriers to mixing. So in the case of the Miramichi and Broad Oar and St. Lawrence populations, they're not really mixing at all. And you can see that again, you know, with the Canadian populations. I'm going to fast forward not too far. where you have this really great separation. So this northern group here, they, by all extent, Canadian researchers think that there's a cold water current off the north side of Newfoundland that cuts them off, sorry, the north side of Nova Scotia, that cuts them off from migrating south. There's been a lot of tagged fish from all over Canada, and there's a lot of tagged fish in the United States. There's extensive acoustic telemetry arrays. We don't see a lot of interchange, if any. I think that we've had one Canadian fish on the DMF arrays over all our years, 10 plus years of running these things, almost 15 now, or maybe more. Bill could correct me if I'm messing this up. And we don't see a whole lot of mixing between these either. The St. John River is almost a putative population. It does seem to be successfully spawning with native fish from there, but it does get also some fish from the US that seem to be coming in, and that's the most connectivity. You can see us closer to the United States here, and that's just going up the Bay of Fundy. This Shubenacadie River population is super cool. They spawn in the tidal bore way up in the Minas Basin, and that seems to be how their eggs stay out of the salt wedge. They really seem locally adapted, and so it makes sense again that they might not be having a lot of mixing with these other populations. So that's kind of, I think, some context that helps with your question, Khalil.
It does, and it's interesting because regarding the Chesapeake, I can't speak for the Hudson. We have a lot of abiotic factors and biotic factors coming into play with the spawning biomass. We do have a strong spawning biomass. They are laying lots of eggs. They're being fertilized. But we have the abiotic factors, the siltation, the fertilizers, the warm water, therefore low oxygen content, affecting the viability of the egg or the fry. You got the biotic factors, the blue-nosed catfish, the snakehead. I think I'm pronouncing that particular invasive species correctly. We've got a lot of different things happening in the Chesapeake that is creating havoc as far as recruitment's concerned, and I'm beginning to wonder whether or not that northern strain, the Canadian strain, the Canada strain, not Canadian strain, but the Canada strain will become the dominant years from now. I'm 82, so I'm going to be gone. But it'd be interesting to see if that becomes the dominant breeding ground for Atlantic striped bass. I mean, it's a fascinating phenomenon that is occurring right before us, and it'd be interesting to see if there are any kind of projections, doomsday projections as to when the spawning biomass can no longer support a species, as far as in the Hudson, and then, in the Chesapeake, and then we're starting to see things happening in the Hudson. So it's a dynamic condition that's occurring before our eyes, and I find your research very fascinating, the DNA research, and it's just interesting to hypothesize as to where this might go in the future.
Yeah, it absolutely is, Khalil. And I think for a couple of years now, I've been in a camp of, and talk to people about, over the, time span of centuries. I don't think that Chesapeake productivity is getting better. It's probably already gone down and will continue to go down. The Hudson, whatever happens there, just doesn't have the potential, the productivity potential that the Chesapeake does. And so, really the only place that does is the St. Lawrence, and even then it may be less. So that kind of, I've been saying, looks like the future for striped bass, and that if we really want to have a lot of striped bass or something comparing to what we have now, we should be thinking about restoring some of these spawning areas that we've lost over time, like it's occurred in the Kennebec, and maybe will happen in the Penobscot, but the Connecticut, the Merrimack, other larger rivers where we might get some production out. And if we removed these first dams on those rivers, or the first couple dams on the rivers, then that might be a way forward too. But given the modeling that came out about the AMOC a couple of weeks ago, maybe on that same time scale, we're just actually going to get colder and locally some of those damages, especially the temperature related ones, will be reversed. So the climate change thing is highly unpredictable, and we'll see where it goes, but certainly doesn't hurt to be proactive. And I think that restoring rivers for all diathermous fish is a really worthwhile goal.
Well, as you know, being a diadromous, one of the diadromous experts on the fisheries. Dam removal obviously is the way to go, but as you know, that can be a 15 to 20 year project that you just don't take dams down just like that. And not you, I mean, but folks just don't take dams down. You can say we want the dam to come off or come down, but it's all the legalities of doing it. There are a lot of roadblocks, and that's you're talking 10, 15, 20 years for a dam removal. But I'm just hoping that we can save this species, this Atlantic states fishery, and that it doesn't go into a demise. So it really is an interesting phenomena that we're experiencing, all of us, and there are no easy answers. And now we have freshwater, excuse me, fishing magazines. Proponents now are advertising striped bass charters in the lakes for the freshwater striped bass, and that's a new phenomenon that is occurring. Guides are taking up guiding for striped bass in lakes in the Carolinas and I think in western parts of the states, and I hate to think that it's going to be going to that. Hey, Ben, thanks a lot. Great presentation.
No problem, Khalil. Thank you.
Thanks for letting me editorialize.
Well, I was going to say, I see a lot of Chesapeake Bay folks here. I'm happy to make this a safe space for people who want to talk about Conowingo.
Well, we're past 12 o'clock. So we going to wind this up. So if you have any questions for Ben, send him an email or give him a call. So I just want to thank Ben again for presenting, and I'll let everyone know of the next talk for next month. So I'll send that out soon. So thanks everyone for coming. Have a good rest of your day.
Thanks, everyone.
Yeah, we'll put a clap here. There we go. All right, everyone. Bye.